we will read them afterwards. So, thank you.
Thank you very much for your kind words and for giving me the stage. So as Nicolas said, I'm currently a sales manager at 3D Ceram, but I used to work on the machines that we have here as an engineer for the best part of 4 years. So hopefully if you have any technical questions as well, I'll be happy to answer them at the end of the webinar. Just for today, so I'll be giving a quick introduction to 3D Ceram and the technology that we use here, and then going through 3 different study cases of ours tied in with Biomedical. So we have our skull implants, which is a very strong part of, a big part of the history of 3D Ceram. We have a research study that we conducted with the laboratory in Lyon on cerium stabilized with zirconia. And then we have a more industrial project that we've been working in Australia on spinal cages. So to begin with a quick introduction to the company. The company first of all started off as a service provider, and then as you can see through the timeline here, we became more of a process provider, changing from providing parts into providing machines and providing complete solutions. So now we boast quite a range of different machines and technologies to answer all of our customers' different needs. Process provider, what does this mean? It means that we provide all the equipment, so the different machines ranging in size. We have the cleaning solutions, but also the firing solutions. We've been working on our own software and extensions. We work on formulations, commercial formulations, but also developing with research, universities, institute centers, etc., like I'll be showing later on, and our dedicated services to make sure that everything goes smoothly. Moving on to the technology we use. So 3D Ceram uses an SLA technology, so top-down stereolithography. So it starts with having a computer model that we then want to print, preparing the platform in itself, and then we move on to the printing of the part. So we use a low viscosity slurry that we spread across the build platform. That we then use a 405 nanometer UV laser with a galvanometronic head to direct the laser across the build platform to print the parts layer by layer. So we chose a low viscosity formulation for many reasons. One being for simplicity in cleaning and also reducing recovery time. So as you can see here with this example of the lattice part, at the end of the printing run, we can raise the platform and we can see the slurry flowing out, which means at the end, we can then print some very nice lattice structure parts. So that was a quick introduction to the company, but getting on to the main topics here. So one of the very first projects that 3D Ceram was involved in back in 2005 was a study that was conducted with the local surgery here at the CHU Limoges to produce 3D printed skull implants. So as you can see here on the picture, the advantages of additive manufacturing were to create 3 different zones on one part, which the part was tailor-made for the patient. So we have a first zone, which is a very dense zone, which to have some strong mechanical properties. We have a second zone, which is more of a porous zone, which was to allow for blood vessels and osteointegration. And then the third are these small holes around the edge, which are actually just for the surgical procedure, which we'll come to later on. So I made sure I put some warning in because there are some surgical photos throughout the presentation. I just wanted to make sure to give everyone a chance to look away if they don't want to see them. So here we actually have an interview with the surgeon that was head of the maxillofacial surgery at Limoges Hospital. And so if I'll just let play the video. So that was just the head surgeon going through the kind of procedure and the outcomes that they managed to see on different patients. There are still some more surgical photos. I'll let you know when you can look back. So one of the first examples was this this patient here, so who has an orbital frontal defect. And so what would happen was there would be, through various scans, taken and design the part that would be printed. We would then go for our printing stage here, go through the necessary certifications to be able to produce these parts that would then be implanted into people. It would go through surgery, and then we can see this is 12 months after the operation, that although you can see the outline, you can see that there's no gap between the skull and the implant, and also that the fact that the implant itself, which is accredited to the HAP, has a very similar texture and color on the scan compared to the original bone. The second patient here had quite a traumatic skull injury, where quite a large section from the front of the skull had to be removed during the operation. And so again, for this purpose, the implant itself was cut into 3 different parts to give some more flexibility during the surgery. So the surgery went very successfully, and then we can see here on the scan afterwards. This was a few weeks after the surgery. We can see how the part is fitting nicely into with the rest of the skull. This last one was also a fairly large skull implant that had to be used. So again, it had to be divided into 3 different parts for more flexibility during the surgery. So the surgery went very well, and then as you see afterwards, there was a lot better results from the study and all. In total, there were 15 different patients that received 3D printed skull implants that were all performed at the local surgery here. So moving on to a different study, which was a more research-based study that we conducted with the National Institute of Applied Sciences in Lyon, so based on cerium-stabilized zirconia. So zirconia with 3% of yttria was one of the first zirconias that was used for orthopedic implants because of different properties that the material had, such as biocompatibility, wear resistance, good mechanical strength, and toughness. However, there is a problem with using the zirconia in hip implants, for instance. This was stopped back in 2001 due to some breakages and damages. What would happen was that the zirconia would age, which was accelerated by the humid and pressurized conditions being inside the body. And so it would change, the structure of the material would change to a monoclinic structure. And monoclinic would mean that the part itself would lose some of this mechanical strength. So what we considered was that after 20% of monoclinic transformation, the part would need to be replaced. So as we can see here, when you extrapolate at 37 degrees Celsius, it means that the biomedical implant could last anywhere between 6 and 60 years, which obviously in biomedical terms is not great, because this means that the patient would be at risk of repeat surgery, which is what we're trying to avoid. So there are several different approaches to try and solve this issue using the zirconia. One would be to replace the yttria stabilized zirconia with zirconia toughened alumina, but there was still the potential aging issue. The other was to go down a doping avenue and dope the zirconia with cerium, so in 10%. This meant that the aging was a lot better with the material itself. However, because cerium has bigger grains than yttrium, it meant that the mechanical properties suffered as a consequence. But in terms of aging, we were now looking at more at 800, 1,000 years. So in biomedical terms, no risk of repeat surgery. So what happened was, it was a collaboration of 3 national laboratories, where the study was headed by Jerome Chevalier and Helen Reveron, where they were developing a new ceramic for orthopedic applications that exhibited plastic deformation before failure. And the idea as well was to have equivalent properties that were obtained with the 3D printing of SMA, as you would with conventional methods such as cold isostatic pressing. So the laboratory itself was coming up with this cerium stabilized zirconia powder, and our part in the collaboration was to come up with a way to be able to use it, to be able to 3D print it, and so offer tailor-made products like we did with the HAP skull implants. So 3D Ceramic, we offer 3 different solutions for the consumables on the machine. The first one is the standard. It's powdered organic materials that are sourced by us. We develop the parameters and then we can give it to our clients. The second one is a more interesting one, which we'll call the on-demand one. So this is where a customer comes with us with a specific powder they want to use, and then we develop a formulation and the parameters to use on the machine. And the last one is on the machines, we have open parameters, which is ideal for research institutes that want to develop their own stuff. So in this case, we were on the on-demand kind of setup. So we took the powder itself from Intervion, was developed completely in their laboratory, delivered to us, and then we would work on making formulation and then working on parameters for them to be able to print. So why we got told they would chose the cerium stabilized zirconia against the yttrium was obviously the mechanical properties and the hardness was reduced, but the aging and the toughness was much better. But because we want to be able to use this in surgeries, it meant mechanical properties still needed to be there. I think to begin with, the powder was supposed to be used in jaw reconstruction. So obviously you still need some mechanical strength. So to kind of balance this out, what happened was the powder had aluminum nanoparticles that were added as a second phase to increase the mechanical properties. And to kind of get this right compromise between mechanical properties and toughness, there was a third phase, which was with aluminum strontium platelets that were added. This meant at the end, we had a new ceramic matrix, a ceramic composite, based off a cerium stabilized zirconia matrix, but with aluminum particles for mechanical properties and aluminum platelets for the toughness. So once with this powder, we made the formulation, we made some test parts, and so we printed some different beams for testing the mechanical properties. We used 2 different sintering temperatures just to see how much of an effect a 50 degrees Celsius variation of the sintering temperature had on the results. With the results that we obtained, so we had a Weber modulus of 16, which was very interesting for us because it meant that the defects were uniformly distributed across the bars, which meant that the printing was very homogeneous across the build plate and the parts were repeatable. And what we can see here is basically that the properties obtained by the 3D printed parts and the conventional methods part differ ever so slightly, but insignificantly when we're looking at sintering with the second temperature sintering. What we can also see is when we were looking through under the microscope for very similar microstructures. So you can see in the different parts that were obtained, the 2 different sintering temperatures, but also the parts obtained by conventional methods, that we could still find the matrix, we could still define where the particles were and where the platelets were. But when we were looking at the parts themselves, when we look at the defects, the tolerance towards defects that ceramics have, with classic zirconia stabilized with 3% yttria, we have a critical defect of 1 micron. With this new composite, we had a critical defect of 100 microns, which meant, as we can see on this sintered sample here, there was a certain flexibility that the part was able to have before breakage. So having a Weber modulus of 16 and critical defects starting at 100 microns meant that there was this more defect tolerant ceramic that was made. And so what we could take away from this was a very good collaboration between us and the laboratory to develop this new ceramic with plastic behavior for the biomedical applications. We could also verify that 3D printing meant that we could obtain the same properties as with conventional methods, but it opens up the advantages of additive manufacturing. With conventional methods, we kind of compromise on the flexibility of design to be able to just print one design quickly and efficiently. Having this increased critical defect meant that we could change the design and be able to be more flexible without necessarily compromising the quality of the parts. And the last study case I have, which is more of an industrial study case in itself, is with a partner in Australia printing intervertebral cages. So Allegro Orthopedics, they're a company that is based in Sydney, Australia, founded mainly with expert designers, engineers, And they've been working in the healthcare sector, mainly on innovating and developing different products such as prosthetics and devices. One of their new materials that they're developing was actually originally developed in the University of Sydney, and it's called strontium hardystonite garnet. I will not be repeating this name many times during the presentation. I will call it SRHT garnet. This calcium silicate-based ceramic, they've decided they're going to be using this as a cervical fusion cage in spinal surgeries. What does this mean? It means for spinal cages, at the moment, they're currently made in mainly 3 different materials: ceramic with silicon nitride, polymer with PEEK, and titanium for metal. But with the current spinal cages, these require an autograft, which means that the patient has a second surgery to remove some bone, generally from the hip. And so this goes into the hole, as we can see on the design of the part here, to allow for the cervical fusion to happen. With the new material, there's also the possibility of having a new design. So as you can see here, there's the idea with this new material is to avoid having the autograft. So in 2021, Allegro completed a large animal pilot study, which is going to be paving the way towards their FDA approval. And then when we look at the market as well, why they were interested in these spinal cages, one was obviously because of the size of the market itself, which is only going to increase because of the aging population, but also the increase in the number of spinal sports injuries. But why to aim for this particular material and to avoid the bone grafting is because bone grafting for spinal fusion accounts for more than half of all bone grafting procedures. So to be able to save so much time, resources, effort, and discomfort for the patient, to remove this necessity to have a bone graft whilst having a spinal fusion. So they actually conducted 2 different studies, which I'll be talking about here. One was on the material itself, so just on the powder. So the objective was to determine whether the material could be used for load-bearing applications. So what they did is they produced some samples just by pressing, because obviously it's easier to do just when you've got just the powder, and make some simple test parts. They tested the physical and chemical properties. They also did some biological tests as well in the simulated body fluid. They also then performed mechanical properties on some more complex parts, on the part, sorry. And then there was some more biological in vitro and in vivo different studies that were conducted. So to begin with the microstructure of ceramics, there's a first type, which is the crystalline ceramic, which fails under loading. Why? Because there's normally some porosity, and with this porosity, cracks then propagate along the boundary of the grains. Second kind of structure of ceramics is having this liquid phase sintered ceramic. Here there's an improved strength, but the toughness is compromised because the cracks will then propagate through this glass phase instead. So what they were trying to do with this new material was to improve the scaffold strength, but also enhance the toughness by using these sub-micron crystals that would help to deflect the path of the cracks. When we look at the SEM examinations comparing the TCP with the new material, you see there's very similar porosity afterwards. The cracks, and you can see the cracks and the defects in the TCP, but you can see with the new material there's a solid continuous microstructure all throughout. And we can see where in the TCP and HAP that there's some grain boundary defects, whereas when you look at the new material, there's a very wetting amorphous grain boundaries, and you can just about see the dispersed submicron crystals to help deflect the path of the cracks. So to begin with, they did some mechanical testing of the solid samples. So you can see with the compressive strength and the fracture toughness. Sorry, I just have a box to move. And they also did some mechanical testing of the porous samples. So what we can see is that the compressive strength is comparable to the cancellous bone. So the cancellous bone is the porous bone that's in the middle of a human bone. Whereas the fracture toughness and the elastic modulus are more comparable to the cortical bone, which is to say the solid, dense exterior to a human bone. And we can see in terms of when we're looking at the cancellous bone, that hydroxyapatite, for instance, does not have the compressive strength that's similar to cancellous bone. We're almost 10 times or 100 times worse off. What we also can see is when through this different study was that under the stress strain curve that we can see on the figure E, we can see there's a very high toughness, but there's also a slight elastic behavior at the end. And we can also see, mainly on figure G, that soaking time influences very little of the mechanical properties, which means that once the material would be implanted in the human body, it would still retain its properties. Another warning, there are some more surgical photos after this. Look away now. So there was a study that was conducted with some radio implants in rabbits. So what would happen with these scaffolds, they were implanted inside a critical size defect, which was around 13 millimeters. And what we could see from this was there was a superior bridging of the critical size defect for the new material compared to TCP and HA after 12 weeks, which we can also see in the radiography here. Then they also did some tests with the interaction of the material with primary human bone cells. So looking at the proliferation and the osteogenic gene expression. What they found was that the osteoblast can detect the osteoinductive signals from the new material, which means that this also explains the bridging superiority compared to the other material. And there's also confirmation of in vivo biocompatibility and osteoconductivity, which means that this material could be used in implants. To take away from the study results, so they were able to make a ceramic scaffold with 85% porosity and 400 to 500 micron pore size, very similar to bone structure. Good compressive strength and modulus. It was biocompatible in vitro. There was no significant deterioration in simulated body fluid. The scaffolds themselves are bioactive, and it was proven to be more capable than TCP and HAP to repair the critical size defects. So this meant it had the potential to be used in load and partial load bearing sites in orthopedic and trauma applications. Which means we move on to the second study they performed. Again, there is a surgical photo afterwards. What they were trying to do was determine the systemic biological safety of this new material as an interbody fusion device. So this was an assessment over 26 weeks of the sheep under GLP, using parts that we had printed with their powder on our machines. So they were looking at all the different biological effects. And as we can see here in the picture, this was the implantation of on the left is just the spinal cage, and then on the right was an extra device that was added on top to keep the vertebrae in place just for the test. What the study showed was that the implants were very well tolerated by the sheep's body. So we can see on the radiographs there's no abnormality. We can see that the articles are very well placed. You can see here where the screws and the metallic fixture is, and you can see where the spinal cages are in between the vertebrae. Throughout the study, the sheep gained weight or had a stable weight, which meant that the surgery did not cause them any harm or did not make them ill at all. And we can see there was no infection by controlling the levels of hemoglobin and also the white blood cell counts, which obviously would increase if there was any infection that they were trying to fight off. We could see that there was a good blood clinical biochemistry parameters. They remained stable throughout the implantation. The strontium concentration was not affected by the presence of the test article. It's important. The strontium in itself inside the test article is what's going to help the bones fuse together without having to use the autograft. But ensuring that the strontium then doesn't become wayward inside the sheep's body is also very important. We saw there was no evidence of risk associated with the incorporation of the strontium fusion cage, and so it was deemed the material was safe for ACDF surgery. This also meant that the investigation of the new material into spinal devices for humans has been supported. And so the next step for them is to obtain FDA approval, and then we're going to help them proceed with human clinical trials. Thank you very much. If you have any questions, or if you'd like to, any more questions, or if there's even the scientific articles that you would like, please don't hesitate to get in touch.
Thank you very much, Charlie, for this very interesting presentation. So we will do the questions and answer after the second presentation. So please write your questions for Charlie in the chat. So now it's my pleasure to introduce my colleague, Damien Coibion, from the Greenmat laboratory at the University of Liège in Belgium. He's also using the 3D ceram technology to print bioceramics. So Damien, the stage is yours.
Thank you, Nico, and thank you to the whole committee of YCN for the invitation to this webinar. Before to start, I would like to give a few words about the Greenmat, which is the lab where I'm doing my PhD thesis. We are working mainly on developing materials for renewable technologies, such as the production of materials for photovoltaics panels, but also the storage of the energy made by this technique by developing materials for batteries, and also developing materials for bioceramics applications. We are also involved in the recycling of end-of-life photovoltaic panels, batteries, but also food waste that I used in my PhD thesis. So, which is about the recycling of bovine bones to recover deoxyapatite, to use it as a raw material for stereolithography in bone grafting. So for introduction, I will give you a short summary of the QN graft techniques and to place my works between them. So the first one, which is considered is the gold standard, it's autograft. So as Charlie mentioned in his presentation, we require 2 surgical operations, which is pain and risk of necrosis at the donor site, even if there is no risk of rejection, and we have the best osteogenic behavior. The other possibility is the allograft, where the bone pieces for the graft is coming from a donor. We still have good osseointegration and conduction, but we have a lower osteogenic behavior and a higher risk of rejection and transmission of disease. Another possibility is the xenograft, quite the same as allograft, but the bone is coming from an animal. We have the same advantages and disadvantages, the higher risk of rejection and the transmission of disease, which is called zoonotic disease. And the last option is to use a synthetic substitute. We have lower risk of disease because of no organics and no body receptors. And we have the possibility to design by 3D printing, for example, the graft to better fit with the defect. And in the drawbacks, we have a lower biocompatibility, mainly with metal and plastic implants, and a risk of inflammatory response. So my work is between these 2 last options, because I'm using bovine bones and to take it as a raw material for the 3D printing, and as it is done in a synthetic substitute. So we are going to take the advantages and decrease the disadvantages of both of them. We have the good biocompatibility from the deoxyapatite coming from the bovine bones. So we have a good osteocompatibility, and we have a lower risk of rejection than with allograft or xenograft, because we are removing the organic parts of the bones, and we have still less transmission of disease. And as I said, I will use the hydroxyapatite as a raw material for the 3D printing, which allow us to design the implant. So what are the advantages of the biogenic hydroxyapatite compared to a synthetic hydroxyapatite? It's mainly the substituent element found in the natural hydroxyapatite. Each site of the unit cell can be replaced by different elements. Mainly the calcium can be replaced by other cations, such as magnesium, sodium, potassium, and so on. The phosphates and hydroxide groups can also be replaced by carbonates, silicates, chlorides, and so on. And it is known that these elements are involved in the osteocompatibility of the graft. Most of them are implied to favor the osteoblast and osteoclast activities, which are the cells involved in the bone turnover and bone healing, and favor the densification of the graft. And carbonates and silicates, for example, are also known to enhance the collagen synthesis. Collagen are mainly is the main organic component of bones, and also will favor and promote the osteocompatibility. So the 3D printed has offered to have the possibility to custom the implants from the medical scan. So it is possible to modelize the required implant to fit the bone defect, and then to have a model file that can be read by the printer and that can do layers by layers the required implant for the graft, and then it's the job of the surgeon to do the graft. So why did we choose the stereolithography? It's mainly for two reasons. The first one is its high resolutions, which is mainly defined by the spot size. Here we have a 35 micron spot size of the laser, and by the slice thickness, we are working with 50 to 100 micrometers of layers. And also it allows to produce as well as small and large parts from few millimeters to dozen centimeters that can fit with the defect. So the bone composition is mainly of two parts. The mineral part, as I said, it is hydroxyapatite, and the organic part is mainly collagen. The 10 remaining weights percentage are mainly water and other pure organics. So we have to remove the organic parts to avoid the rejections by the body receptors and to avoid transmission disease. And to do that, we have mainly two possibilities. We're using two different techniques. The first one, as I can say, it's the conventional techniques, which is the calcinations, where the organic molecules are decomposed into CO2. In the literature, we can find temperature between 700 to 900 degrees C, and the dwelling time are most often between 4 and 10 hours. So we can recover an organic-free hydroxyapatite with a quite low specific surface, close to 5 square meter per grams. The other techniques is a technique that we are developing for the moment in our lab, which is called the oxidative hydrothermal. So we are able to degrade the organic matter into oxidized water-soluble molecules, and we are doing it at lower temperature compared to calcinations, so between 200 and 300 degrees C. And we can do it without dwelling time or with a dwelling time of 2 hours, so in a shorter time also. So we recover an almost organic-free hydroxyapatite. We have less than 1 weight% of organic matter inside. But compared to calcination, we recover a high specific surface area, close to 70 square meter per gram. So as you maybe know, for the stereolithography, because of the incompatibility of the hydroxyapatite with the resin, we need to have the lower specific surface area. So that's why I'm working with the calcination technique. And the other techniques is used to prepare powders for water treatment, which is another part of my PhD thesis. Because of its high specific surface area, it allows to have a better contact with the water and improve the extraction of the waste in the water. So after the calcination, we have to deal with the particle size and morphology. So we are doing it in 2 steps. The first one is ball milling. So we are working with a suspension where balls are hitting the hydroxyapatite particles and are going to reduce the size of the particles. So at the end of the process, we have a quite narrow distribution of size with a mean diameter of 1.7 micrometer. And after that, the suspension is dried in a spray drying chamber. So we have a spray in micro droplets that are drying thanks to hot air that is going through the drying chamber, and we recover the powder. So what is interesting here, we can see that after the drying, we still have quite the same distribution in size with almost no modifications. We still have a mean diameter of 1.7 micrometers. As you can see on the same picture, we have aggregates of these particles that are easily break when we are preparing the paste. And we have, as I said before, a specific surface area close to 5 and 6 square meter per gram. So the next step is the preparation of the paste. So we have mainly 4 components inside the paste. The first one is the recovered hydroxyapatite, and the other main component is the resin. We are using an acrylate resin. And as I said before, there is quite incompatibility between the hydrophilic hydroxyapatite and the hydrophobic resin. So we have to use a disperging agent, which is in our case phosphate esters. And we are also adding a plasticizer, which is the type of polyethylene glycol. So to disperse and to have an homogeneous dispersion, we are using a dual asymmetric centrifugal mixer. And after that, we put the mixture in the 3-roll mill in order to mill the remaining agglomerates and recover a smooth paste. And at this stage, we have to see if our paste have the good behavior, good rheological behavior. So to do that, we put a few paste inside the rheometer between 2 plates. The bottom one is fixed and the upper one is rotating. And during the rotation, which is increasing, we will have a look at the viscosity. As you can see, at the quite steady states, we have a very high viscosity. And when we are increasing the rotation and that increase the shear rate, we can see that the viscosity is decreasing. So we found the shear thinning behavior required to have a good spreading of the paste inside the 3D printer. So the next analysis that we are performing on our paste is the 3-interval thixotropy test, which is used to simulate the scrapping. So in this graph, you can find 2 sets of data. The first one, the red one, is the storage modulus. We can say also the solid modulus. And the blue one, which is the loss modulus, can be compared to a liquid modulus. So the graph is separated in 3 zones. The first one corresponds to the steady state of the paste inside the 3D printer. There is almost no shearing on the paste. And what we can see is that the storage modulus is above the loss modulus. It's almost 6 times, I think, higher than the liquid modulus. So at this stage, we have more solid state of the paste. So the paste will not flow by their own on the plate. Then on the second part of the graph, we are applying a shearing state, shearing corresponding to the scrapping. So at this stage, we can see that the liquid modulus is above the storage modulus, the solid modulus. So here we have a good spreading of the paste on the plate. And when we stop the shearing, we can see a really fast recovering of the better solid state compared to the loss modulus. So as we compare the ratio between the solid modulus and the liquid modulus before the scrapping and after the scrapping, we can see that we have an immediate recovering of 72% of the initial ratio, and we have to wait only 32 seconds to have the full recovering. The next characterization that we performed are tests of polymerizations. So we are printing only one layer by one, and so we can measure the thickness of this layer compared to the density, the energy density that we are applying on the paste. So the energy density is mainly dependent of the time and the scanning speed of the laser and its power. So we can plot this kind of graph with the curing depth is a function of the density energy. And from this graph, we can find some information. The first one is the penetration depth. So it is the penetration of the laser until it has 37% of his initial energy, and also the critical energy density, which is the lower energy required to print our paste. About the penetration depth, we have quite the same value compared to the literature, which means that the particles are not deflecting the laser, and we have a good penetrations. But about the critical energy density, it's quite low compared to the literature, which means that our particles are not absorbing the laser, and the laser can directly polymerize our resin. So thanks to this graph, we can also control the energy required for the desired curing depth that is required for the printing. So that's what I do for this part. So we are printing geared-like pieces. So for the depositions, we have layer thickness of 100 microns. So we decided to have a curing of 200 microns to have continuum in the layers that can stick together. And to do that, from the previous graph, we determined that the power of the laser required is about 60 milliwatts, and the scanning speed is about 5,000 millimeter per second. So here are the first results. So you have the green body, so the has printed part, and after sintering, of course, we have a shrinkage of the pieces of almost 40% in volume, I think. It's close to 15% in each dimension. And you can see on the same picture that after the debinding and the sintering, we have a quite good resolutions. For the debinding, we work with a temperature ramp of 20 degrees per hour. And for the sintered, we have applied for 2 hours a temperature of 1250 degrees C. So I would like just to present what I would like to do in the next steps. So in next steps, I would like to compare different structure. So quite simple cubic unit cell with diamond unit cell, but also more stochastic unit cells such as Voronoi samples, and compares the structure, the influence of the structure on the medical resistance and also on the cell adhesions. So I decided to have similar properties than the cancellous bones. So as presented, Charlie is the part which is porous in the bones. So we have quite a lot of voids because we have a pore volume of 75%. The size are close to 300 micrometers, and the wall thickness are about 150 micrometer. So I hope that I can present you fast these results in maybe in the next webinar or another meeting. So thank you for your attention. I will be glad to answer to your questions if you have.
Thank you, Damien, for this very nice presentation also. So now is the time for questions for the two speakers. So if you want to put your picture, you can use the chat and I will read the questions to the speakers. I think we have already one question from Islam for Charlie. So he's asking, so you've conducted any post-op follow-up. Could you share the infection rates you have observed? And additionally, could you briefly revisit the rationale behind choosing specific porosities in craniofacial graft?
Yep. So in terms of post-op, from what I know personally, there were no infections following the surgery. In terms of long care post-op, at the moment I don't know. We did an interview with Joe Bree a few 3 years ago now. We talked about it. There's a lot of different things he goes over. I think the video is on the company's YouTube channel if anyone's ever interested. The idea between having the specific porosities was for the osteointegration. So as we could see on the scans afterwards, the implants fused well, allowed the blood vessels that were around the skull to integrate into the implant itself. And that's how we can see that how we managed to have skull implants that had very little gap with the skull itself. How everything stayed in even 12 months after when the scans were made.
I think you answered properly. So thank you, Charlie. We have a question now for Damien. Did you perform any CFD calculations? So I guess it's for fluid simulations. Fluid dynamics were done before printing the porous scaffolds.
No, for the moment, and no calculation were performed, but maybe that's something that can be done in further time. Yes, maybe.
Okay, thanks. There is another question for Damien from Mateusz. So he wants to know which atmosphere you used for the dividing of your samples.
Yes, we use for the moment air, but I know that in some publications are using nitrogens. Maybe that we will compare the impact on maybe crack separations or something like that in next experiment.
Yeah, about that, I have maybe just curious questions. Have you thought about the alternative dividing options like microwave, CO2 supercritic or something?
Yes, that's something that I'm thinking about, but we do not have the equipment in our lab. But maybe if there is possibility of collaborations with another lab to do microwave sintering, that's something that is interesting me. for the advantages of fast curing also. So yes, that's something interesting.
Okay, thank you. Yeah, there is a comment from Wadi for Damien. He said all the best for your next investigations, and he sent you a link to a paper that did a similar investigation, and they showed that the diamond TPMS structure was the strongest.
Thank you, because it's always interesting to have comparisons with the literature. So after my test, I will compare with their results.
Yeah. Thanks, Wadi. There is a question for Charlie. So your second and third materials were zirconia and strontium granite. And what was the first one?
Hydroxyapatite.
Yeah, I had a question for Charlie for this new material, the strontium HT granite. So they use it with strontium, but is it possible to, I don't know, use another type of cation or dopant? I don't know, like magnesium, or I don't know if you know.
No, quite possibly, but the powder itself was developed at the University of Sydney, and then Allegra acquired the rights to be able to use it, and then started the original scaffold study to check that the material was usable. And then for the ovine study, that's when they asked us to be involved to be able to produce a formulation with the powder to use on the machines here. So we printed some samples for them for the study. And pretty soon again, we'll probably be working with them and producing some more samples for them for their next study, which will be in the human clinical trials for the FDA approval.
Okay, I see. Thanks. I don't know if there are any more questions from the audience. I may have just one other question, maybe for Damien. So you said you don't use the powder you got from the hydrothermal treatments because it's like the surface specific is too high. What would be the solutions to be able to use these powders for the printing process?
A quite easy possibility is to do a thermal treatment on the powder to decrease its specific surface area, but we will maybe lost the benefit of these new techniques to recover the powder, and we can do the same with the calcinations, which is quite direct. But what can be interesting with the powder recovered from the hydrothermal treatment with its high specific surface area, it's maybe not possible to use it for paste preparation in stereolithography, but in powder techniques of additive manufacturing, maybe we can use it in binder jetting or fuse deposition and so on, powder bed fusion, sorry. Like that, we can use this powder for this kind of applications.
Okay, thanks. I don't see any more questions in the chat. There was Evan Kumar. He had something for Charlie, but I don't see the question.
Maybe he's still writing.
It's typing.
Yeah, maybe it's still writing. In the meantime, I just had also a question about the sintering of this from the study of INSA Lyon for Charlie,
Yep.
because they use like different materials in the parts, like alumina and this cerium zirconia. So are you aware of any sintering issues they had with these different materials in the same part?
No,
because they need to be compatible for the densification.
no. From what I heard, because it wasn't like it was a composite, like a multi-material kind of print. Because all of the blending had been done at the powder stage, everything was very well dispersed.
Yeah.
From what I heard from them, they didn't have any issues with the sintering. But again, it might be because they possibly didn't share the information with us on the. But no, from what we had, they were able to sinter the parts, and say with the microstructure SEM views, we can see that the microstructure is very similar to what they obtained with conventional methods.
it looks like more additives than completely multi-materials. Yeah. Yeah. Good. Oh, there is. I don't see any more questions from the audience. So just again, the webinar is recorded. It will be available on the YouTube channel of ESERS on YouTube. So everything will be there, the recordings. So now we have questions for Charlie. So have you determined toothing transformation in ceramics TZP?
So the tetragonal zirconia powder.
Mhm.
That one I'm not too sure, to be honest. Not too sure. With the study, it was defined as the transformation from quadratic to monoclinic was the reasoning behind the loss of mechanical properties. But to answer your question, I'm not too sure.
I think the team of Lyon they published these papers and they studied
So
maybe the transformation. So maybe you can find more information in the papers. Yeah. Maybe more conventional parts.
Yeah. So the paper's published on the names of Sylvain Fournier, Jérôme Chevalier, and his team.
Yeah, thanks. So if there is no more questions, I would like to thank again Damien and Charlie for their time and to share their research contributions. It was again a great webinar co-hosted with the Bioceramics Network. So thank you again. And again, the webinar will be online on the YouTube channel. So please have a look if you missed some of the information. So thank you for joining us and have a great afternoon. And keep in touch with the YCN and the Bioceramics Network. And if you can contribute to these networks, it's always a pleasure to receive nice contributions.
Thank you very much.
Thank you again.
Bye.
Thank
you.
Thanks,
Bye.
speakers. Thank you.
Bye.
Bye. See you soon.
And Charlie, nice to see you. See you now remotely, but still. I know we discussed.
You too, Aga. How have you been?
I'm fine, thanks. I'm fine.
Yeah.
Bit the flu season still ongoing, but with the small kids, this is how it is.
Yeah.
Part of the deal.
Yeah, I know.
Thanks, Damien. Nice talk. And I have to say, we do something similar with those substituted hydroxyapatites and try to print it. So interesting work, and it's a real challenge, of course.
Thank you. Yes, it's interesting to see other techniques of substituting or doping. So why not use the natural hydroxyapatite that already has all these elements? So we are trying to use it.
And when do you think you'll be able to try the cubic, the Voronoi, the diamond-shaped kind of structures?
I expect that yes, maybe I also working. I didn't speak about that, but also with the geometries of the scaffolds, so cylinder or hexagonal parameters.
Yeah.
So maybe that the angles will favor the cell adhesions, but also something that will maybe favor rapid cracks on something else. So I think that the cubic will maybe favor the cell adhesions and the diamond also, but the cubics will quite fast as we have lined already prepared for the cracks. In the diamond structure, yes, we will maybe more disperse the energy and have lower not easy cracks. And Voronoi, the stochastic structure, which is the most similar to the real bone. Maybe also we'll have both compromise with quite good saladations, because all of the tortuosity inside the structure and quite a good dissipation of the forces inside the piece because of different directions of dispersion of the force. Maybe it's my expectation, but the science will prove or not these expectations.
And what about gyroid?
Gyroid, I think that at the Biocam conference, yes, it was 2 months ago now.
Yeah,
or 1 month ago.
December, yeah.
Yes, I saw that there are a lot of works of gyroids which can favor the infiltrations of the cells. So it could also be a quite interesting structure. I just have to modify the current piece that I have, because they are not similar to the one that I prepared about the pore volume and the pore size and so on, to be comparable with the others. But it could also be a quite good structure for the grafting.
Yeah. Okay. I look forward to seeing your results.
I hope that I will produce it quite fast.
Okay, guys, thank you again.
Thank you very much.
Always a pleasure to keep in touch.
Yes. We are not so far from each other, Nico.
No. Unfortunately,
Europe is small.
Charlie and
We're all quite close to each other.
Yeah, for us too, we have a bit more traveling to do.
Mm-hmm.
Okay.
Okay, guys.
Have a nice day. Have a nice weekend.
Take care.
Thanks.
Again, thank you.
See you again. On the meeting.
Yeah.
Bye, guys.
Bye-bye.